L'altoparlante vicino al microfono.
Ok.
Allora, posso accendere questo?
Se dovete parlare entrambi.
Sì, no. Design synthesis and biological evaluation of new inhibitors of carcinogenic processes.
Good afternoon.
You have 15 minutes,
Yes, okay.
and I will be very strict because of the time is not enough for every speaker.
Good afternoon to everybody. My work is focused on discovery of a new potential drug as a strategy of cancer therapy. Cancer is the result of uncontrolled cell growth. Cancer cells, in fact, still share many of the same needs and property of normal cells, but they are out of the normal control. The process by which normal cells switch into abnormal cells can take a long time. In fact, the tumor progression is a stepwise process, where the development of blood vessel is an important step. In fact, cancer cells produce migratory growth factor and proton conjugate protein. These proteins are involved in the signal transduction pathway that play an important role in the tumor progression. It is important for the transmission of the signal from the outside environment through the cell membrane into cytoplasm and nucleus, where the transcription contributes to generate this protein. These proteins are vital for the cancer cells. In fact, they are involved in processes such as proliferation, invasion, metastasis, and apoptosis. All of these proteins are correlated with HSP19. In fact, this protein stabilizes these oncoproteins, and the inhibition of this protein degrades this protein and stops the tumor growth. The complex array displayed for cancer cells can be organized in 6 different traits. This includes the ability to evade apoptosis, the ability to be self-sufficient for growth, the ability to metastasize and undergo unlimited replication, the ability to promote neogenesis, and the ability not to respond to the anti-growth signals. All of these features are correlated also with HSP19. In homeostatic condition, HSP19 is a molecular chaperone, and it is involved in folding, biological function, stabilization, and activation of the protein. This protein, called the Klein proteins, includes the protein kinase such as RB2 and AKT, the cyclin-independent kinase, the CRAF, IF1-alpha, and the mutant p53. These are vital for the cell life. In an environmental stress condition, such as heat, radiation, reactive oxygen radicals, toxin, epoxy, or bacteria and viral infection, we have an overexpression of this protein to launch a rapid and protective response against the external insult, without a need to going to otherwise replication or transcriptional period. This protein comprehends 3 different domains: the carboxy terminal domains, which it is important for homodimerization and co-chaperones recruitment. A middle region represents the bind site for Klein protein, and it is important for the ATP activity, and the amino terminal domains, which represent the bind site for ATP and for the inhibitors. The known HSP90 inhibitors include natural products such as geldalamicin or radicicol, the semi-derivative synthetic semi-synthetic products such as 17-DMEG and 70AG. They are currently in second phase on clinical trials in USA and the United Kingdom. Purine compound or pyrazole compound. Thus, it is easy to understand how this protein can be considered as a potential target for cancer therapy. So we have characterized the pocket protein which bind the ligand by hydrophobic interaction and H-bound acceptor and donors. In the left image, we have inhibitors of HSP90, geldalamicin, in the pocket protein. And the same interaction were found for our most active compound on the right imaging. By a molecular docking and pharmacophore approach to analyze the subset of 5 million of compound available in zinc database, we have designed and selected 112 compound as potential HSP90 inhibitors. Among them, we have selected and synthesized the compound which present the aminocyano-pyridine core structure, the pyrazole, and the triazole pyrimidines compound. About the synthesis, the chalcone 8 was obtained in two steps. This is the common intermediate for both the pyrazole and the aminocyano-pyridines compound. For the triazole pyrimidines, they were synthesized by a 1,3 dipolar cycloaddition reaction. These present a planar structure, so they can enter in the stacked base phase nucleotides of the DNA, and moreover, if they present suitable chain, there are further interaction with the marrow groove of DNA. Some of these heterocycle compounds were selected for biological screening in National Cancer Institute Developmental Therapeutic Program. In this anti-cancer screening, the compounds were tested again as 60 cell line panels, which belong to 9 different tumor types. Among this compound, the derivative 16 was the more active. In fact, it was active in leukemia panel and in central nervous cancer panel, and above all in the renal cancer, where it presents a growth inhibition 50 around 8. So this compound is active at 10 nanomolar concentration. Also, the compound 10 was tested against the 16 cell lines at a single dose 10 micromolar. So it was active above all in the colon cancer panel. To summarize, we can say that HSP90 represents an important target for cancer therapy. In fact, the involvement of this protein in acquisition and maintenance of tumor phenotype is particularly interesting, and suggests that the inhibition of this protein can be correlated on all key processes on which the cancer cells based for their growth and their survival. Thanks for your attention.
Very on time. Thank you very much. Any comment? Any question? Short comment or question? Thank you very much.
Thank you.
It was very interesting in this field, very important for our